Okay, so today we're going to talk about muscle contraction. So when we talk about muscle contraction, there are a lot of things that comes to play, and the thing that needs to come in our mind is fiber, muscle fiber. So today we're going to talk about what happens in the muscle fiber to cause contraction. But before then, we would like to talk about some of the terms that we normally come contact or we normally see when it comes to muscle contraction. So the first one is called sarcolemma. So sarcolemma, when you take a fiber, every fiber has got an outer membrane or a cell membrane which covers it. And this is called a sarcolemma. So it happens to be the first part which interfaces with a neuron whenever there is any transmitter or a neurotransmitter being transmitted from a neuron to the fiber. So there is the first part that interfaces the neuron. And just take it as just an outer membrane or any protein membrane that or cell membrane that covers the muscle fiber. And on the sarcolemma, there are some tubes on the sarcolemma, and these tubes are called transverse tubules. We will talk about that one, transverse tubules. So through these tubules that when a neurotransmitter is being transmitted from a neuron to the fiber, it passes through these tubes and gets into the endoplasmic reticulum, which stores calcium in the muscle fiber. So we also have another term which is called sarcomere. So with sarcomere is just the smallest functional unit of a muscle fiber. Every basic thing, everything that we have, everything like let's say a molecule has got atoms, a building has got bricks. The basic unit of a building is bricks. So the same thing when we take a muscle fiber, the smallest unit in a fiber that you can talk of is called a sarcomere. And this sarcomere is where contraction happens because it has got these prominent proteins acting in myosin, which is the protein filaments that are found in the fiber. And these are responsible for contraction in the muscle fiber. We will talk about them, their functions later. So the T-tube, as I said, the T-tube is a tiny tube-like extension of the sarcolemma that goes deep into the muscle fiber, and its main job is to carry the nerve impulses, that's the action potential from the surface of the muscle, that's the sarcolemma, into the inside of the fiber. And it allows a signal to reach each part of the muscle at the same time. So just take it like the brain wants you to raise your hand and or wants a particular fiber to contract. The brain transmits message through neurons, and every neuron has got an axon. At the base of the axon, we have some synaptic vesicles. They are like sacs, which contains some chemicals, and these chemicals are called acetylcholine. So acetylcholine, they are the neurotransmitters or the chemical that contains the messages which the brain wants to transfer. So when these vesicles burst, it releases this acetylcholine, and then it will now be transmitted through the synaptic cleft, which is the space between the axon and the sarcolemma. It will pass through these tubules and gets inside of the fiber. There is a particular smooth endoplasmic which contains calcium. So when these transports or when this action potential gets inside of the fiber, it tells the sarcoplasmic reticulum to release the calciums. So we will get there. So as I said, sarcoplasmic reticulum, these are just a smooth endoplasmic reticulum which are found in the muscle cells inside the fiber. Its main function is to store the calcium and release the calcium as and when it's needed. So when a nerve impulse, which is being transformed from the brain through the neuron, through the synaptic cleft, through the T-tubule and gets into the sarcoplasmic reticulum, it will tell the sarcoplasmic reticulum to either release the calcium. Then we also have other proteins which are found in the sarcomere. We talk of the actin and the myosin. On the actin, we have other proteins that are regulatory, like on and off, which tells the other proteins to how they should act. So these two proteins are called troponin and tropomyosin. We also learn about that one as well. So let's look at this particular function. Let's look at this particular structure. This is a sarcomere. Let's look at this fiber, marks fiber. This whole thing that you see here is a marks fiber. There is an outer membrane which is called a sarcolemma or sarcolemma, and this is what covers it. And it is this sarcolemma which has got some receptors on it, which is called acetylcholine receptors. They receive any impulse or potential that comes from the neuron, and then they will transmit through the T-tubule, that's transverse tubules, and tell the endoplasmic reticulum to release the calciums. And when these calciums are being released, they interface or binds with the troponin, and then the contraction starts to take place. So let's take this whole thing as a fiber. When you take this whole thing as a fiber, you can see from here that the fiber contains microtubules, like smaller sub-units of the fiber, which is called as microtubules. And when we take just one thing of the microtubule, is called a sarcomere. That's the smallest unit of a fiber. So when you break it down a fiber, the last objects or the last thing that you see is the sarcomere, which contains the actin and the protein. Okay, so we said when brain wants to transfer or give any message to any part of the body, it passes through the neurons. And neurons are their function is supposed to transfer messages from the brain. So if the brain wants you to wink, the brain wants you to speak, the brain wants you to laugh, the brain wants you to do anything, it gives signals through these neurons, and the neurons will act accordingly. So every neuron has got an axon. And when you look at this, these are the axons. It has a lot of axons. So when we take just one particular axon, the base of this axon, that is what is being drawn here, very big here. And the base of the axon, that's called a terminal. That's the ending of the axon. Inside the terminal of an axon, it has called synaptic vesicles. Like they are like sacs. So these are sacs, and you can see these green stuff inside the sacs. They are called acetylcholine. That's the ACH. Acetylcholine. They are chemicals. Even when you hear the name acetylcholine, it's a kind of some carbon, chloride, and other chemicals combined. And these are neurotransmitter. When you say neurotransmitter means it's a chemical that contains a message. So it's just a transmitter. So neurons, transmitting message. So it's what it carries is just a message from the neuron, and it's just transmitting. And these are all the neurons. And you can see this axon and this particular another neuron. Between a neuron and a neuron, there are some spaces. They don't touch. And the space between the neuron and another neuron is called a cleft. That is synaptic cleft. The same thing when the space between another one neuron and a microfiber is also called synaptic clefts, because they don't touch. They need to be able to release this acetylcholine to whichever receptor that they are supposed to bind to. So synaptic vesicles, just see that when you see vesicle means a sac. So they contains those chemicals, that's the neurotransmitters. So whenever a neuron wants to transfer message to any part of the body, it gives an impulse through the neuron. And when this neuron arrives at the axon terminal, it makes the vesicles burst open, and it releases this acetylcholine into the synaptic cleft. That's the space. And then you see this ligand-gated channels, which will also try to absorb this and bind to this and now move on to the next step. When we talk of acetylcholine, as I said, it's a neurotransmitter, a chemical that carries a message from the nerve to the maxilla. This acetylcholine, when it is being released, these are the small dots. They are contained in the sac. So when an impulse comes and the sac breaks, they are being released through the clefts here into the space here, and it diffuses across the synaptic clefts and binds to receptors on the sarcolemma. So we will see the receptors. The receptors on the sarcolemma are like some white shade, and is always looking forward to bind to an acetylcholine. So we will talk of that one. So synaptic cleft, as I said, is there just a tiny gap between a nerve ending and axon terminal and the maxillary fiber membrane, sarcolemma. It is where communication happens between nerve and the maxilla. So at this particular junction, this whole thing is happening where acetylcholine is binding to receptors and communication from an axon is connecting to or getting to the maxilla. That junction is called neuromuscular junction. So neuron and the maxillary junction. So we call it neuromuscular junction. That is where this whole chemical binding and everything takes place. And one thing is when it happens between a neuron and another neuron, we don't call it neural junction, but that one we call it synapse. So the junction between one neuron and another neuron where communication takes place or where these acetylcholine are being transmitted to is called synapse. So a synapse is a junction between two neurons where a signal is transmitted from one cell to the next. And this communication occurs through either the release of chemical messengers, which are the neurotransmitters, or using a direct electrical signals, that's electrical synapse. So there are these neuron or neural maxilla. So there are differences. If it is between a neuron and a neuron, it's called synapse. But if it is between a neuron and a maxilla, it's called neuromuscular junction. So as I said, this is, let's take one neuron and this is a maxilla. And these are synaptic vesicles which contains the acetylcholine. So if acetylcholine is being released into the synaptic cleft, which is the gap between the neuron or neoaxon and the maxilla, there are some receptors. The receptors are like white sheet. They will be receiving this acetylcholine. So once these acetylcholine are being released into the space here, they need to be found somewhere. They need to be binded to something. And these neural acetylcholine receptors will bind to this acetylcholine. And then it will now try to pass through the T tubule and go inside the fiber. So the junction where all these are happening is called a neuromuscular junction. So now we are now going to dive deep into the muscle contraction. So we talked of the sarcomere. Sarcomere, we said the sarcomere is the basic units of a muscle, or let's say the smallest unit. When you want to break a muscle and break it, the last one that you see is sarcomere. And sarcomere is a protein filament. It has a lot of proteins that helps in the contraction. There are two main proteins that are responsible for contraction in the sarcomere. One is called actin and one is called myosin. When you hear the name actin, actin is very thin. It's a thin protein filament. But the myosin is a bit thick than the actin. The actin is very thin, but myosin is thick than actin. Then there are other proteins which are responsible for regulating whether contraction should occur or should not occur. So they are termed as regulatory proteins. So those controllers, they are the troponin and the tropomyosin. And all these proteins, these two proteins, the troponin and tropomyosin, they are found on the actin. They are responsible for regulating how contraction should occur. by controlling actin and myosin interaction. And there are other proteins which controls the elasticity and the alignment, how a muscle should stretch, how a muscle should be maintained, the alignment of it. These proteins are called structural proteins. They are all found in the sarcomere, as we talked of. An example is the titin, nuclein, dystrophin, desmin, etc. So let's take actin. So we are now talking of the sarcomere, and the sarcomere, one of the proteins that is responsible for contraction is called actin. And the actin, when you look at the actin has is a thin strand of protein or filaments, protein filaments. And you can see it's made up of a lot of globular units or globular actin. So these are like subunits. They are like spheres chained together. And on top of each of these, there are some dots on them. And these black spots are called the binding sites. They are the binding sites on the actin. And the binding sites, what are they used for? We'll talk of them later. And on top of these binding sites, you can see there's another thin filament covering these binding sites. So it's like the binding site is waiting for something to be bounded to. But when you look at this, there's something covering, so that means nothing can bind to unless this thin filament is taken off. And the thin filament that is covering the binding site is called troponin. But for the tropomyosin also to cover it, there are some other proteins that is holding the tropomyosin in place so that it will not be taken or pulled away. And so you can think of it as a staple or a peg or something. So when you dry your clothes on the dry line, you put pegs on them. You peg them so that whenever there is a wind blowing somewhere, the things will not be taken away. And troponin is let's say the staple that is stapling the tropomyosin in place so that will cover all the binding sites of the actin. So this is actin. Actin has got those two regulatory proteins, the tropomyosin and the troponin. So that is actin. So this is also this was just one piece of an actin just showing. So here this is also like the full piece of an actin. So you could see it's a long strand. It's just not this. So you can have it like how our DNA structure and everything looks like. The same thing that is on the fiber. Also, this actin also looks like. So it has got a lot of spheres chained together. That has got a globular unit, and these ones has got binding site. The binding site are being covered by the tropomyosin, and the tropomyosin are being held in place by the troponin complex. That's the troponin protein. So then how will contraction occur? Because for contraction to occur, we need to pull away the pegs so that the tropomyosin, that when I talk of pegs or staples, I'm talking of the troponin. You need to pull away the troponin, and the troponin will also then pull away the tropomyosin so that the binding sites will be exposed. So once the binding sites are exposed, then the myosin can now attach to the binding sites on all these for contraction to occur. So for contraction to occur, myosin needs to bind to the actin for contraction to occur. So that's how contraction occurs in our muscles. So we will come back to that one later. So let's move on. So myosin, this is myosin. This is also one of the protein filaments in the sarcomere that is also responsible for contraction in our muscle. So for the myosin, it's just a long strand of protein and with a head and with two binding sites. One binding site is for actin. That is, there's a part that it will bind to the. It will be used to bind to the actin, and this part it will use to bind to energy. That's ATP, adenosine triphosphate. So let's look at it this way. It has got a head. The head has got two binding sites. One is used for binding to actin and one is used for binding to energy or ATP. So how it does is when ATP is energy. For a muscle to move and bind to actin, it needs energy. So the energy that's ATP in our muscle, energy in our muscle is called ATP. ATP needs to bind to this ATP binding site. Then when it binds to the ATP binding site, this myosin will now be energized to get the energy and to try and pull itself to go and bind to the actin binding site and contraction occur. So that is myosin. So this is how contraction occurs. In your brain, it will send a nerve signal. As we talk of a nerve message will come from the brain and pass through the spinal cord and to reach the axon. But then it will pass through the neurons through the axon. When it gets to the axon, let's look at this one. So this is your axon terminal. The brain will transmit an impulse, a nerve impulse, pass through the axon and gets to the axon terminal. The axon terminal contains synaptic vesicles. These synaptic vesicles contain acetylcholine, which are neurotransmitters, which carry messages from the brain. So these vesicles will break. When they break, they pass through these spores and get into the synaptic cleft, into this particular space. When they get into this space, they need to be received by something. And this is your muscle. The maxilla has got a thin lining on the maxilla, which is called a sarcolemma. Sarcolemma too has got receptors which are Y-shaped, and these Y-shaped are called acetylcholine receptors. And these will receive the acetylcholine. Once they receive them, they will now pass through these T tubules. These are the tubules, and these T tubules will pass through these T tubules and diffuse into the inner part of the fiber. When it gets to the inner part of the fiber, there is a smooth endoplasmic, which is called a sarcoplasmic reticulum. It contains calcium. The moment it gets to this particular sarcoplasmic reticulum, this SR, it directs this particular SR. This SR contains calcium in our muscles. This is a place where muscles are, calcium are being stored. So once this acetylcholine gets into the muscle and directs this sarcoplasmic reticulum, it will now tell this SR to release its calcium. Once the calcium are being released, when we come to the sarcomere, so the sarcomere contains actin and myosin proteins. And we saw that the actin has got troponin, which are like staples, which binds the tropomyosin on the active binding site of the actin. So once the calcium are being released, it binds to the troponin. So let's look at this again. It binds to this troponin. And once the calcium binds to this troponin, the troponin will now change shape. The moment it changes shape, it's like you having a rubber and subjecting it to heat. You see that the rubber will change shape. The moment it changes shape, it will now pull away the long, thin filament, which is the tropomyosin, away. The moment it pulls away, the binding site on the actin will now be active, or will now be exposed, looking for something to bind it to bind to. And then once it becomes exposed, then energy or ATP will bind to this ATP binding site will bind to myosin head here, and then myosin will now be energized. So now we have the actin binding site exposed, waiting for something to be bounded to. So now ATP will bind to myosin head, and it will get the myosin energized. The moment ATP binds to myosin and gets energized, the adenosine triphosphate gets the myosin energized. Then it will now try to pull toward the actin binding site, and it will try now to bind. So you can see this is myosin and this is actin, and the head will now try to bind to the active site or the binding site of the actin, and it will try to pull the actin and itself towards the center, and this will cause contraction. So this is how the whole muscle contraction is being done, or how it occurs. So acetylcholine release from axon terminal binds to receptors on the sarcolemma, and action potential is generated and travel through the two tubules. Now it tells the sarcoplasmic reticulum to release calcium, and this calcium binds to the troponin. And when they bind to the troponin will change shape and it will pull the tropomyosin away from the actin binding site. Once they are being pulled away, the actin binding site is exposed. Now, ATP, which is energy, binds to the myosin head, causing it to energize. Once it causes it to energize, it will now try to bind to actin binding site. And once it binds to the actin binding site, it will try to pull it into the center and now causes contraction. The moment myosin binds to actin, the place where it is occurring is called a cross bridge, because this is one protein, this is the other protein, and then they are crossing. So it's like a bridge where they are meeting, and it is called a cross bridge. So when we talk of cross bridge, is where the myosin and the actin, when myosin and actin binds together, that is what we call a cross bridge. And this thing can repeat as many times, and it always repeats in the body. That's how it's been done. So once another ATP binds again to the myosin head, it will now cause the myosin now to detach from the actin. Once it detaches, you see that the troponin and the tropomyosin will now come back to its place again, and the binding side of the actin will be covered, and the calcium that was used to bind to the troponin will now be absorbed back into the sarcoplasmic reticulum, and now the myosin also will now also pull back, and now relaxation of the myosin will also occur. So it's like just a reverse. So that's how this muscle contraction occurs in our body. Thank you for your attention.